Our next speaker is Heath Blackmon.
Okay. First, I'd just like to thank the committee for giving me the opportunity to talk about my work today. I'm going to talk about the fragile Y hypothesis, which is my attempt to formalize the role that Y aneuploidy or Y chromosome loss has in sex chromosome evolution, genome structure evolution, and even the evolution of meiotic mechanisms. So why should we care about Y or W chromosome loss? If we just thought about vertebrates, we might not think it's that important. We would see a couple of losses in rodents, a handful in fish, and that's about it. But over the last few years, I've spent a lot of time as part of two projects collecting all the karyotype and sex chromosome information that we can on invertebrates. And I now have records for about 15,000 species. And what we see is across all invertebrates, about 16% of them lack Y or W chromosomes. So these are XO or Z0 species. And for the rest of the talk, I'll talk about XY systems, but most of what I'm discussing would apply to ZW systems as well. And even in species like our own, where the Y chromosome is required, oftentimes the lack of a Y chromosome is a common disease condition called Turner syndrome. And this happens when we have a failure during male meiosis to pull the X and Y chromosome to opposite poles, and they both go to one pole, this Nondisjunction event. And so I think this is really exciting, and talking about it here is especially fun because you've seen a couple of times this first issue, the first article in Genetics, and these non-disjunction events that Calvin was using to look at the chromosome theory of heredity, these are just these events where the X and Y chromosome both went to one pole, and so the parents were contributing either two sex chromosomes or no sex chromosomes to the offspring. So it's kind of nice to bring it full circle, even if I have no chance of competing with that hair, because that's like some amazing hairstyle there. So what I want to do is just start off with like the traditional canonical story of sex chromosome evolution. We're starting out with two autosomes that are identical. We have the evolution of some sex-determining locus. And then there's this idea that sexual antagonism is going to drive this divergence between the X and Y chromosome. We're going to have a gene that makes you a good male, and we want to link that together with the gene that makes you a male. And so oftentimes this is going to lead to the fixation of inversions, where we link more and more of the Y chromosome together in this non-recombining region. And we expect this to happen cyclically, and I'm showing the Y chromosome shrinking there because once it no longer recombines, things like Muller's ratchet are going to take effect and start reducing the genic content of that Y chromosome. And when you read the literature, what you'll see is they'll say that this process continues until the par becomes small, and that there's a limit there because the par is required for proper segregation. And so this par is just that region at the bottom that I'm showing, and that's where the X and Y during meiosis in males is going to come together and has to physically link together to hold the chromosomes together until it's time to segregate to the poles. But the thing is, when we look at pars in the few species that they've been well characterized in, we see that there's actually a tremendous amount of variation, about a 7-fold variation just in these mammals in the size of par regions. And so this really we should wonder whether or not this variation is important. And I would argue that we can actually look at our own chromosomes and human data and see that there's a hint that this variation may indeed matter. So these plots, what I'm showing you, on the x-axis, we have the largest chromosome arm for each chromosome in our genome. And then on the y-axis, I have the aneuploidy rate. And this is measured in blastocysts and also in sperm. And we can fit a simple linear model, and what we find is we can explain about 20 to 40% of the variation in aneuploidy rates just based on the size of the largest arm of each chromosome. So our large chromosomes are going to be down here in this right quadrant, where we have the big chromosomes and low aneuploidy rates, and then our smaller chromosomes are going to be up here, where we have less room for the chromosomes to come together and pair, and we have higher aneuploidy rates. So if we think about how this would apply to sex chromosomes in males, where these par regions are so essential, we can imagine that par size now would be on the x-axis, and when we have a young sex chromosome that has a large par region, we're going to be down here in this quadrant, and so there's a large region that can come together and pair up and hold the chromosomes together during meiosis. But as chromosomes age, we're going to end up here in this part of the graph where there now is a very small region for the chromosomes to pair. And so if we think about how this would work out as an evolutionary process over time, we can imagine that we're going through this transition in par size. And I would say that as that par size reduces, we're going to have an increasing rate of aneuploidy. And once we get out here to an old sex chromosome with a small par, we now have the strong selective force that's going to be pushing to resolve and bring down that aneuploidy rate and increase the fitness of males. And so we can imagine three ways that this might be resolved. One would be fusions or translocations from the autosomes onto that par region that would expand it back out. That would move us basically back in time in this graph to where the aneuploidy rate's low again. The second is we could just evolve a new kind of meiosis that doesn't require a par. So we could evolve achiasmatic or asynaptic meiosis where a par region isn't required to properly segregate the X and Y. And then finally, we can move those few genes that we have that are on the Y that we need. They could be moved to autosomes or to the X, and then we can lose the Y at no cost. So what I wanted to do with this was actually look kind of across the tree of life and see if I could see signatures of this force that we would expect to find. And I came up with two predictions. One is that when we evolve one of these alternative segregation mechanisms, like achiasmatic meiosis or asynaptic meiosis, we should see a drop in the rate of Y chromosome loss. And then the second is that species with small par regions, they should lose the Y more often. So if we think about where we can test this, we might look at mammals, and we could compare metatheria and eutheria, and it would be consistent with our prediction, but it's also not very convincing just because of the size of the groups. We could have the exact same rates here and still see this pattern. So we need a better group to look at, and I would argue that beetles are the place that this can really be tested powerfully. So I made this time tree that lets me look at about 1,000 species of beetles, and the reason they're so powerful is these two suborders that are on this tree. I've painted Polyphaga to be blue and Adiphaga to be red. They have completely different types of meiosis. So in Polyphaga, we have the asynaptic meiosis, which is where the chromosomes, sex chromosomes in males come together, but they never pair up with each other. Proteins hold them at a distance until it's time to segregate to the poles. In contrast, in Adiphaga, we have largely chiasmatic meiosis, just like our own, where there's a par region that pairs up. And in both of these groups, we have over 100 species that have already lost the Y chromosome, and these are spread out across the tree, so we should have enough events that we can actually get a good estimate of how quickly this is occurring. And so I fit an MK model that just has a specific rate that you can estimate for how quickly we're making this transition. And the rate estimates that we see are consistent with our prediction. The group that has the asynaptic meiosis is losing Y chromosomes much, much slower than the group that has chiasmatic meiosis, where we depend on this par region. One problem with this analysis, though, is that, again, with beetles, suborders are looking at very distantly related groups. So it could be that the sex chromosomes in these two groups just have different genes that's changing the kind of constraints that they're under. So we'd like to look at maybe a lower level comparison where the changes are more recent. And it ends up that this one suborder, Adiphaga, offers this opportunity. We have two small clades within it that have recently evolved achiasmatic meiosis. And so what we can do here is I use an approach called PPS, posterior predictive simulations. I take this tree that I have, and then theta there is my model and the parameters that I've estimated from it. And I just simulate new sex chromosome data starting from the base of my tree up to the tips. And what this does is it gives me this distribution of expected numbers of Y chromosome losses, and then I can compare that to what I actually observe in the data, which is the red line. Now, if our prediction that achiasmatic meiosis reduces Y loss, we should see this red line, the empirical, should shift over to the left relative to our simulations. And so when we look at these two groups, this is exactly what we see. In this first group, Trechety, we see 3 species that lack Y chromosomes, but 95% of our simulations have more than that. And the second group is the same way. We only see 1 Y chromosome species that lacks a Y chromosome there, but 92% of our simulations have more. So in both of these groups, we're seeing a reduction in the number of Y chromosome losses relative to the rest of the suborder. The second thing that we'd like to look at is par size. Unfortunately, par size isn't characterized well, so we're going to use a correlate for par size, which is chromosome number. So if we think about across a large tree of species, the way that chromosome number changes, fusions and fissions are going to be two of the primary ways. And sometimes these are going to involve sex chromosomes. And so if a fusion occurs with a sex chromosome, we're reducing the number of chromosomes, and we're going to expand out that par region. In contrast with fissions, we're going to increase the number of chromosomes, and if it occurs in that par region, then we're going to reduce the par size. So across a large scale, we should see this negative correlation between par size and chromosome number. What this allows us to do then is we can go back to that big tree of beetles, and we can reconstruct chromosome number and Y chromosome loss. And so on the right, I'm showing you where I've estimated the Y chromosome losses to have occurred, and we can just look up what the chromosome number was at each one of those points. And then we can also create a null distribution by picking those points at random on our tree. And we can do this in those two large suborders, and we see exactly what we would predict, which is an adiphaga where they have this chiasmatic meiosis. The species that are losing Y chromosomes are the species that have more chromosomes, on average of about 15 chromosomes versus the null, which is a little over 12. In contrast, in Polyphaga, the group where we don't have a par region, we actually see no difference between where we see Y chromosomes being lost versus the null distribution. And then also we can just look at the patterns that we see in eutherians, and we see this really interesting clustering of outcomes. So those three things that I predicted to happen once we get these old small par regions, Y chromosome loss, achiasmatic meiosis, and fusions, we see all three of those happening in Muridae, in closely related genera. And then in Chrysetidae, the other group that has Y chromosome losses, we also see the evolution of achiasmatic meiosis there. When we look across the rest of eutherians, we don't see those, although we do see an occasional sex chromosome autosome fusion. So in conclusion, I would just say that Y chromosome loss is much more common than is widely appreciated, and we need to think about it more. And that loss is more common when meiosis is chiasmatic. Loss is more common when that par region is small. And what I think is really exciting about this is that this provides an explanation for some things that either I think were poorly explained in the past or were always assumed to have one explanation. So, for instance, autosome sex chromosome fusions, this has largely been explained through sexual antagonism. But I would say that also simply having high aneuploidy rates could favor fusions between autosomes and sex chromosomes. And what's especially interesting here is that we see an excess of fusions between Y chromosomes and autosomes. And that's the type of fusion that would reduce the aneuploidy rate under this model. And so that's consistent with our results. And then achiasmatic meiosis is another really exciting one, in my opinion, because previous explanations for achiasmatic meiosis were that it's the same pressure that's driving reductions in recombination, and yet we see it evolve out at the tips of the tree with species that have very old sex chromosomes. And in those cases, achiasmatic meiosis isn't going to resolve very much standing sexual antagonism, so it makes much more sense to evolve instead as a response to these high aneuploidy rates, I believe. And I'll take any questions. Oh, actually. And I'm going to end the same way that Calvin Bridges ended, which is to be continued, because that 51-page article was just the first half of that article. The next issue had another 20 pages in it. Because now I'm actually measuring aneuploidy rates in sperm across several mammal species, so we'll be able to have some hard evidence on this in the near future. And so I just would thank my current mentors, Yaniv and Emma, the University of Minnesota, and the University of Minnesota for giving me money to do this sperm analysis on mammals. And then Jeffrey Damuth, who was my PhD advisor and listened to me talk about beetles for far too long. So I'll take any questions in the minute that I have left.